All right, welcome back, guys. We're going to go ahead and get started. I got a few announcements to make there before we begin, though. Number one, the crime scene investigation. I will release the information regarding what happened on that tomorrow, so you can look forward to that. If you haven't gotten it done yet and you want to still do it, you can do so. If you don't want to do the spoiler alert, you just don't look at the document that I post tomorrow. Come up with your motive, figure out what you think happened and why it happened, and then you'll see it from there. And again, that's kind of like what forensics people would do in that field of investigation, trying to figure out what happened, piece together the pieces, and go from there. Sometimes they're right, sometimes they're wrong, but a lot of times what they're looking for is motive and also probable cause of why they would do what they do. So sometimes the stories can get complicated, so you just have to see what happens tomorrow. Number two on the list today, the Chapter 9. We are done with that. We're moving on to Chapter 11. So I will be posting the Chapter 9 quiz tomorrow as well. You can take that anytime you want. There is no due date on that. That can just test your knowledge and see how that goes. Okay. So again, just give it a look, see what you think, and go from there. So test your knowledge. Again, I really appreciate you guys tuning in today to ask questions, to see a live lecture. Even if you're not tuning in right now, that's fine. You're watching the video later. Thank you for tuning in. I'm glad you are taking the initiative again to further your education. That's what it's all about. Okay, so what we're going to do is go ahead and get started with the presentation here. I want to make sure that the document is ready to go. It looks like it might be a little laggy today. Something's up. We'll try. So you can always follow along with the notes online. I posted those, but it looks like the live is a bit behind for some reason, but we'll go from there. All right, well, I'm going to start on Chapter 11 notes, mitosis, and get into the cell cycle as well. So here we go. If the video is not playing along, like I said, just follow the notes. Chapter 11, mitosis, is another little short segment that I like to talk about in advanced biology just because it is important that we go over it. It's something that you will encounter in your college level biology course. Why do cells actually divide is the question, and that is to maintain homeostasis, and that's, again, encompasses a lot of things to keep the cell healthy, but mainly it's the surface area to volume ratio. So when we're talking about the surface area to volume ratio, we talk about why cells have to be so small. So the surface area, think of it as like the mouth, and the volume is the body of the cell. As the cell gets larger, the surface area and volume will both grow, but one of them will grow faster than the others. All right, the volume will grow significantly faster than the surface area. And as such, it makes it difficult for cells to maintain a large volume. So again, I think I gave this analogy before. Think of this: if you continue to grow throughout your lifetime, and by the age of, say, 30, you were 300 feet tall. That's pretty incredible when you think about it. But let's say the only thing that didn't grow on you was your mouth. So as such, you wouldn't be able to get enough food through that little bitty tiny mouth to feed a 300-foot tall person. So it's very important that, again, you have that surface area to feed it. There's also growth and development, which is, again, a single-celled zygote to an adult. So we're talking about embryonic development, going from that one cell to a multicellular organism as yourself today. Reproduce asexually, the cloning of cells in your body, and reproduction of a single-celled organisms like bacteria and archaea. So even your cells do this, like I said, for growth and repair. Your skin cells, you get a new layer of skin about every 27 days, and as such, I mean, those skin cells are still skin cells, and they're clones of one another, essentially. So your body's actually cloning itself in order to repair itself. Other organisms like bacteria, they do that as a form of reproduction. There's reproduce sexually, the production of gametes, which is egg and sperm cells, which is very important, again, in creating new life for the next generation. Types of cell division. You have mitosis, which are identical daughter cells, also identical to the parent cells. So everything that we're doing in mitosis is cloning, essentially, the cells. Make somatic cells, or all cells except for gametes, used for growth, repair, development, and asexual reproduction, one division of the nucleus. Meiosis has 4 haploid daughter cells, and they're not identical to the parent cell, nor are they identical to each other. Used in animals to make gametes, egg and sperm cells for sexual reproduction. Gametes fuse during fertilization to form a diploid zygote. So basically, you're trying to cut down the number of chromosomes in an egg or sperm cell so that when they fuse together with another egg or sperm cell, that will create a diploid offspring, and that's what you are, is a diploid offspring. Chromosomes, we know, we've talked about those in the past. DNA strands that contain genetic information or genes. Each organism can have a different number of chromosomes. We as humans have 46. Humans' DNA, we have approximately 3 billion base pairs, which is about 2 meters long or 6.5 feet long. Humans have up to 21,000 genes, which is pretty incredible. I mean, we have a lot of different genes, which means we code for a lot of different proteins. And as we talked about before, that a gene can have multiple segments, which can be reorganized with those exons to make multiple different proteins. So again, we're talking about a lot of different proteins embedded within your body, not just 21,000, even more than that. We have condensed versus decondensed. Different stages in the cell's life will result in how compacted the DNA will be. And why is the question? Well, you think about it, if it's all loosey-goosey and we go to do cellular division, as we've been kind of talking about with mitosis, that could break apart. And if it breaks apart, that could cause damage to your genes, which cause damage to you and your cells, which not would be good in the end. That's not going to help maintain homeostasis. Chromosome terminology, a couple things. You have autosomes, which are chromosomes numbers 1 through 22 that code for everything but your gender. So it codes for your eye color, your hair, your bones, I mean, like how the formation of your bones, the formation of your lungs, your heart, all that stuff is all coded for in the autosomes. And that's something that we share amongst each other as, again, male versus female. We all have lungs, hearts, and stuff like that. However, that 23rd chromosome is the sex-determining chromosome, and chromosomes that determine the male and female reproductive organs. That's that genitalia that we've been talking about to produce those gametes, eggs or sperm cells. And we had that discussion in class about hermaphrodites and a true hermaphrodite. Again, usually those individuals that are born with both sexual organs externally don't always contain both internal organs like testes or ovaries. They usually just contain one or the other. And that's usually what helps guide parents to make those decisions, usually at birth when they find that. Chromosome terminology again. You have homologous chromosomes, and the word in homologous, we're talking about the word homo in science, which means the same. Chromosomes inherited from your parents that are similar in shape, size, and genetic information. So the chromosome number 1 that you got from mom is going to be very similar to the chromosome number 1 you got from dad, essentially. They both have something, say, for eye color or hair color written on them. So there are chromosomes out there that we all universally kind of share and where they're located at. Chromatids, one strand of a replicated or duplicated chromosome. So when you think about a chromosome, it's usually just a single strand all condensed down, but it can replicate, and then it becomes that classic X form that we always talk about or see, and you'll hopefully see one here after a bit. A sister chromatid is 2 strands of replicated or duplicated genetically identical chromosomes. All right, a few more little terminology things before we progress forward is haploid versus diploid. Haploid I like to think of as half the genetic information. And it's usually we say half because those is what you find in the gametes of egg and sperm cells, and we represent them with just the letter N. So technically you only need, the 1 through 23 chromosomes to actually really generate a human and all the capabilities. However, you always have a backup set to replace any faulty or mutated genes. So it's always good to have an extra set in humans. So cells that have 2 chromosomes, we call them 2N or diploid. And that's what the majority of your cells in your body are diploid. Your heart cells, your brain cells, liver cells, all diploid, except for your egg and sperm cells. So here we go. This is a chromosome right here. As you can see, the blue one and the red one. They are homologous chromosomes because they are similar in shape because they're very large as compared to these two, which are much smaller. When we look at this left half, that is a chromosome. We have another chromosome there, and then we have another one on the red and here. So it looks like 2 X's, but we actually have 4 copies of a chromosome. So in this picture total, we would have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes represented in the picture. Okay, so pretty big difference. All right, chromosome terminology again. Here we look at the maternal chromosome. Again, you have a paternal chromosome number 1, and you also have the maternal chromosome number 1. So that's one for mom, one for dad. So maternal's mom, paternal's dad, FYI. And again, you contain both of these, and again, that's just so you have an extra copy in case you get a faulty gene. All right, so the cell cycle, just to talk a little bit about this. So you normally have the cell cycle, which encompasses everything that the cell does within its lifetime. Interphase is the longest phase of the cell's life. It's where the cell wants to be at the majority of the time. The reason why is because that's where it's going to do its work. It's going to do its 9 to 5 job, growing, working for you, making proteins, doing all that transcription, translation, just so we can get the proteins that we need. Okay? In interphase, it's divided up into 3 subphases, the G1, S, and G2, in that order. The G1 is cellular growth and normal function. The S is the DNA replication, where the sister chromatids are formed or made. And the G2, the growth and prep for cell division. So we're starting to get everything ready, microtubules, spindle fibers, all that, organizing that DNA down to its chromosome form, getting it all ready to go in the G2. And each one of these has checkpoints. If you haven't satisfied what happens in the G1, you have to wait and fix it before you can progress to the S. If the DNA hasn't fully replicated when it's ready to go to G2, it has to wait. And the G2, same thing, has to wait until before it goes to mitosis, which is the next little piece here. All right. Now the reason why a cell needs to wait is because if it does not hit these checkpoints, it could become cancerous. If it does not meet these things and it cannot fix them, most likely the cell will undergo apoptosis, which means it kills itself. And that's killing itself for the greater good, you. Because if it becomes cancerous, it no longer works for you and could kill you, as we know. So the cell cycle, you start with interphase. You have mitosis, which is the cell division, which we'll talk about today. And then lastly is a process called cytokinesis, and that's just the division of the cytoplasm, dividing it into 2 equal halves, basically the cytoplasm. All right. So that's the nutshell of the cell cycle. I would like to talk a little bit about mitosis and some of the phases real quick. We may not get through all of them today, just a few of them, but would like to kind of mention these as we go because they're also very similar to meiosis phases, in which we will get to those as well. All right, so mitosis, the division of the cell's nucleus or the DNA, and this is again to ensure that you have cells that can be identical to one another in your body and perform those tasks that are given to them, whether it be a skin cell, heart cell, brain cell, that needs certain information to make certain proteins to carry out its everyday life and jobs for you. So we have 4 phases: prophase, metaphase, anaphase, and telophase. Cytokinesis is a process, and it happens kind of somewhat. It can start at the end of telophase, but ultimately it finishes up on its own. So it's after that. So in prophase, okay, sometimes we call them the mTOX. Again, we're just talking about the mitosis stages here, or the microtubule organizing centers, as they like to call them. They are cellular structures that move to the opposite ends of the cell during mitosis for help form the spindle fibers. The centrioles are at the core of the mTOX. So we've talked about those little pieces before. Chromosomes condense down into their sister chromatins. The nuclear membrane breaks down. Now, typically the nuclear membrane is a safe, secure vault that houses all of our DNA. But now we're actually going to break that thing down, and the chromosomes are going to be released out into the cytoplasm for the cellular division. You think about it, the nucleus is that safe, secure vault, but the reason why it breaks down is because it grants it more space to do all this. So more space, less chance for errors or mix-ups and stuff like that. So having this nuclear membrane breakdown can seem pretty risky or dangerous, but at the same time, it's also very important that we do this too. Okay? So that's prophase. We can see the chromosomes, centrioles, spindle fibers starting to form. Everything's starting to gear up for this big phase. In metaphase, we have the chromosomes actually aligned along the equator. The centrioles of the mTOX have made it to the poles. Spindle fibers have attached to the kinetochores and are moving those chromosomes around. Now, here's where it gets cool. You're thinking, how do they actually know? How do they move them, right? Well, if you remember going back and talking about proteins, we talked about those motor proteins. Motor proteins are responsible for moving those chromosomes to align along the center of the cell. And they really do this. You can actually look at them underneath the microscope. And if you remember in biology days, we actually looked at onion root tip cells. And the onion root tip, you got to look at the cells undergoing mitosis. And so you can see some in prophase, some metaphase, anaphase, and telophase. The ones in metaphase were really easy to identify because they literally line up right along the quarter or the center of the cell. And the reason for this is logistics, so that when they actually do pull these sister chromatids apart and then pull them in opposite directions, that they arrive uh, pretty much at the same time at the poles. Because if you had one way down here and is pulling it apart, the one way up here may not make it in time, and the cell tries to pinch together, and now you're lacking an entire chromosome, which is not good. So logistics-wise, it just helps when all chromosomes arrive at the poles at the same time to keep things organized. All right. Again, here though, the nuclear membrane is gone. The nuclear envelope, it does not exist here. All right, so it's completely broken down, giving us more room to get this job done. All right. The next phase here is anaphase. Chromosomes are pulled to the poles by the mTOX again. Sister chromatids pull apart because become individual chromosomes. So again, we still have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. And again, this copy here is exactly the same copy as this. And we did this in the S subphase of interphase. This is the whole DNA replication process that we've discussed previously also in another chapter. All right, I'm going to stop here today since we're getting close to time. But I will finish up with the rest of mitosis next time and start talking about some meiosis as well. So I'm going to leave it up to you to ask any questions that you might have about mitosis or the cell cycle. Again, I will post a discussion board after this video. Actually, I'll do it here in a second, and also a video quiz. They're very short, very easy to do. That's just to keep you on top of things, answer a few questions to make sure you understand the content. So if you have any questions, again, you can always email me. I'll be glad to answer those. And if it's a really good one that I did not address in the video, I'll post that to Google Classroom so everybody can see it too. Okay? I'm going to go ahead and post the assignments for you. And I'll wait to see if there's any questions. Hopefully this information made sense. It's kind of just a review from your biology days. So I didn't think it was too difficult. Usually everybody does well on this chapter. But it has been a while since we've gone over some of that stuff. So that's why I like to go over it, just to make sure that everybody's caught up and on the same page as we progress forward. Alright, so the assignments are posted now. Yeah, give them a look. Again, just check them out and see if you can answer the questions and respond to some of your peers. You guys have been doing a great job on the discussion boards. I really appreciate that. Coming up with some unique answers and also asking some really good questions. So keep it up. Doing great. In other news, I'm going to try to shoot some videos for my environmental science class. It's a little tough to push out some work to them because it's an outside class. So I'm going to try to make some short educational videos that you might see pop up on my YouTube channels. This is going to be the adventures of Kentucky Coffee and Sheila Wallaby, which will guest star me and my daughter. So it should be pretty comical if you want a pretty good laugh and to learn a little bit about the environment or nature. Should be a pretty good time. So look forward to those if you happen to find them.